Well, thank you very much for the invitation to this meeting. It's a change of topic, although it's still biology or connected with living things. Well, my name is Alejandro Monastra. I work in the Universidad Nacional de General Sarmiento. It's a relatively new university in the outskirts of Buenos Aires. It's quite near there. And this work was done in collaboration with people from the University of Buenos Aires, mainly with Luciana Bruno. And she works in the physics department of the University of Buenos Aires, doing biophysics. And so the other people is working in the Department of Biological Chemistry, which were doing all the experiments with the living cells. So I will talk about microtubules. The microtubules are structures inside the living cells, mostly the eukaryotic cells, which have a nucleus. So although there are several filamentous structures inside the cells, we have the actin, mostly muscle cells. There are some intermediate filaments, and then there are the microtubules, which are the thickest structures. They have around 25 nanometers of external diameters. They are done of two alpha and beta tubulin dimers. These are proteins, and they are structures, key structures in the eukaryotic cells. So they have mainly they have a structural function inside the cell, although they play a role in the mitosis, for example, and also in the distribution of cellular compartments. They are also the main structure in cells, in cilia and flagellar. So, for example, like the talk of Rodrigo Soto, for example, in this bacteria, these tails are constituted mainly of these microtubules. And they also are like kind of highways inside the cells, doing a kind of cargo transport function done by molecular motors that I will talk a bit later. So they carry organelles like vesicles and mitochondria from the nucleus to the membrane, or like a highway. So also the microtubules are dynamical structures. They are all the time growing and shrinking, and they are built and destruct in some way by polymerization and depolymerization. And so, of course, this process, sometimes they put some intrinsic curvature in these tubes. And well, as I said, they have around 25 nanometers diameter. They are a kind of cylinder, but a hollow cylinder. So, and they have also an internal diameter around 14 nanometers. And their typical lengths goes in this range. I mean, they could be shorter and a bit longer, but they are around 20 micrometers. So more or less the radius of a typical cell. So here there is a movie done by the collaborators in the biological chemistry department. So this is a film, a movie of a living cell. They put some fluorescent proteins that stacks in the microtubules, and we observe a very dynamical process. These microtubules, they are not straight. This could be done by this intrinsic curvature, but we observe, of course, some dynamics in the tubules, the microtubules. So if you watch some particular microtubule, you can observe some curvatures that they create, they grow, and they decay. So that was the main subject of our study. So these cells are from a frog, from the skin of frogs, that they are very planar. So it's very suitable to do this kind of movies. So they are quasi-2D cells. So these backlinks are dynamical undulations in the range of few microns, so they are shorter than the total length of the microtubules. And we observe that they behave like a kind of semi-flexible slender beams. So if we take some beam of some flexible material, we can flex it. One can think in the first sight that this could be done by Brownian motion or thermal noise inside the cell. But it seems that, I mean, the question is that one can compute this typical decorrelation length that is called the persistent length due to thermal fluctuations. And for the case of microtubules, this is around 3 millimeters. It is much bigger than the size of the cell. So these backlinks cannot be done just by random forces due to Brownian motion or thermal motion. So there should be some active forces doing these backlinks. One hypothesis could be that this process that builds and shrinks the microtubules, the polymerization and depolymerization, could be that in this process they could do an active force. There is also another hypothesis that these forces could be produced by entropic forces, by cross-linkers, that they do not depend on the ATP. ATP is like the fuel of the cell. So cross-linkers are smaller tubular structures that they could attach to the microtubules. And there is the last hypothesis that these forces could be produced by molecular motors in a direct or indirect way. Well, all of these processes are non-equilibrium processes in the way. I don't know. I mean, these processes are done, I think, I'm not biologist, by proteins attached to the tip of the microtubules. And what I know is that this process of polymerization, the polymerization is done constantly. And the process of the molecular motors, of course, these motors, they run by ATP. So this is a kind of artistic picture of a microtubule, and this is a molecular motor. So, for example, one of these molecular motors is done by kinesin. So they are really like structures that they walk. They have like 2 feet. They go step by step along the microtubule, and they attach in the other tip to some organelle that should be carried inside the cell. So, of course, when they are walking, they produce forces that they track the organelle, and also they produce a reaction force along the microtubule. The typical forces of these molecular motors are estimated in the piconewton range. So in the experimental group, I already showed this picture of the big picture of the cell, but they can also make zooms, and they can also color as I said before, by a green fluorescent protein, the microtubules, but with another protein they can color the organelles that are carried by the molecular motors. The molecular motors themselves are very tiny to watch them directly, but one can observe the organelles that they carry. And so, for example, in this other movie, we can see this horizontal microtubule. It has some backlinks at the same time as the organelles are carried. So we don't know if it's a reaction, a force horizontal. There can be also, because they are all crossed with one another, so maybe sometimes there are some organelles that they transverse along a secondary filament microtubule, and so it pushed also so there could be several. The scaling, I think the previous one, yeah, it's around, I don't have the exact from the second movie, but this is the range of 5 micrometers. Sorry? Yeah, it's those, I mean, yeah, it's like 30 micrometers frog cell. I said it's 3 millimeters around. So it's much larger than the size of a cell. So as I said before, I mean, it's not possible that this could be done only by Brownian motion or some random thermal motion, energy. So what we have done we argue that the main hypothesis for these active forces on the microtubules are done by these molecular motors, but the force that they produce can be done in very different ways by secondary microtubules or direct forces. So we did a mechanical model. This is based on the worm light chain model. And so we can start from a continuous theory for the beam, for the filament, considering it as a beam, homogeneous beam, so the shape does not change along the beam. So we have an elastic energy that is given by the strain that could have the microtubule along its coordinate L. So this L is the coordinate along the neutral axis. And of course, we have the bending energy that is the energy given by the curvature of the microtubule. So it's the resistance to flex it. So with this continuum theory, one can compute a continuous force. That this could give us differential equations that usually cannot be solved in the general case. So we discretize the microtubule in n segments. So we have a discretized version of the elastic and bending energy. So we characterize the configuration of the microtubule by the position of n plus 1 positions along the microtubule. So deriving these potential energies and also taking into account that the microtubule is immersed in a very viscous medium. So the cytoplasm is something very viscous. It's around 1,000 times the viscosity of water because there are a lot of complex structures, and indeed it's not an homogeneous viscosity. It could be very heterogeneous. And so from these potentials, we have the forces, we have the viscous force, we have a Newton equation for each element of the microtubule, and we will work in an overdamped regime because, of course, this is a very high viscosity, so we will neglect all inertia terms in the movements of the microtubule. So the parameters that we used for the model were taken from the bibliography. So as I said, of course, these energies depends on the Young modulus. E is the Young modulus. A is the area, the transversal area of the microtubule. I is the second moment of inertia. And EI is called the flexural rigidity because it's the parameter that say to us how difficult it is to flex a beam. So these are the typical viscosity, the diameter of the microtubule, the drag per length parameter C can be computed for a long cylinder. This is the drag per unit length of a cylinder. So it's a very complex, how to say, from Navier-Stokes equation one can solve it. It's a very difficult task, so it's not possible to do it for an arbitrary geometry, but for a cylinder it's estimated in this way. So we put this coefficient in this range. Of course, to the model we have to add the boundary conditions. So to know if the yeah, I mean, this parameter is quite high, so indeed it tells us that it's very difficult to compress or to. Yeah, there are a lot of models that they do like a rigid, incompressible or rigid rods connected, but in these models you have to apply in the algorithm the way to the constraint to have a constant length, and we think that this, because I will show now with some simulations, when you do a compression of the microtubule, indeed you are, even if it's just a tiny bit that you're compressing the microtubule, it adds a lot of energy that then it goes to the bending of the microtubule. So here, of course, we have the constraint, but these are the geometrical constraints given by the situation. And we can add to the model, of course, external forces with different strength directions, and it depends the evolution, of course, where the force is applied. So this, for example, is just a simulation with free boundary condition. We're considering, for example, just two forces in the tips, equal forces. They are bigger than the critical force. So at the beginning, even if you say the straight tubule in this, there is a long time where the instability is growing, but it's not, we cannot see it. But then once the buckling develops, it grows very fast. So this is a condition for constant forces. We also consider a condition where one tip is fixed. Here in this movie, this tip will be fixed, and we will push here with a constant velocity. So, and here some stabilities develop very soon. And we observe similar shapes with the ones in the living cells. So, because of this freedom that we have in the mechanical model, we consider different scenarios for the living cells. So, sometimes this is the schematics of a molecular motor walking along a secondary filament, and sometimes the organelle could touch a microtubule, and then it push it with a constant force. Of course, not extremely constant, but we consider this scenario. Sometimes, if the organelle is attached to something inside the cell and it still walks along the filament, this is like if you walk around a slippery carpet, then you take the microtubule in the other direction with almost constant velocity. And of course, sometimes an organelle can push a microtubule in a lateral direction. And we also studied what happened because these molecular motors, they attach and then detach from the microtubules. So once they are detached, the force stops and we observe a relaxation. Because in this dynamical process, we observe growing the buckling and then it decreases. So in the tangential constant force, we perform the simulation. Here in this picture, what I want to show is if the initial shape of the microtubule is quite straight, we observe that the amplitude, this is the amplitude of the buckling, it grows exponentially. And then it saturates depending on the force that we are doing. On the other hand, if the microtubule was at the beginning already buckled with some amplitude, what we observe is that the buckling develops in a linear way at the very beginning. Of course, the force should be bigger than the critical Euler force. This is just by engineering. When you compress a beam, you have a critical force that depends, of course, in the flexural rigidity, the length of the column, and some parameter, numerical parameter that depends only on the boundary condition. So, for example, in this case, for this 5 micrometer microtubule, the critical force is 32 piconewtons, so we did a force of 40, just slightly bigger. In the constant speed scenario, as I showed in the second movie of the simulation, we have these instabilities, so the shape of the microtubule in the first seconds, or in the first instance, we observe some instabilities, but then very soon the buckling develops more or less with the same shape that in the constant force scenario. Yeah. Yeah. I mean, by action and reaction, we hypothesize that there should be more or less of the same order of magnitude. I mean, you have the molecular motor. I mean, the force is done really on the microtubule because it's walking. It has this system that. And so here we also see that it depends of the initial shape of the microtubule. If we have a very straight microtubule, there is some instant of time at the beginning that's what I said before, that you are compressing the microtubule, and then at some point the backlink develops. And this correlates indeed with a very high peak in the force. So here the velocity is constant, so the force that the motor would be doing, it could be very large. So this is an hypothesis. But as soon as the backlink developed, the force decays to some constant value in the order also of some 40 piconewtons. For a lateral force, the amplitude, final amplitude is, you can solve this even analytically, just doing a force, constant force in the middle of a microtubule with constraint in the tips. So the amplitude is proportional to the force. So more force you have, a bigger amplitude, lateral amplitude of the backlink you have. And for relaxations, here we started with already buckled shape. We take out the force, and so we observe a very rapid decay. So this is the fastest decay. This also can be estimated analytically. The characteristic time depends, again, on the flexural rigidity, on the drag coefficient, because the relaxation, of course, it depends on the viscosity of the medium, and also on the length and some numerical factor, again, depending on the boundary conditions. The question is, if we start with a shape and we relax, but a force that is still bigger than the critical force, the microtubule relaxed to a shape with a smaller amplitude. So in this case, the characteristic time is longer. So we see here that the characteristic time to go here is longer than in the case of just a free relaxation. And well, I go now to the experimental results. So from these movies that I showed, there were several movies of the order of 20, 30 movies, and they observe, they track the shape of the microtubules with a software that it was developed previously by the group, by the experimental group. So after the shapes are tracked even in the time, so in the movie they have a dynamical tracking of the microtubule, and they fit the shape with a Gaussian. So this is arbitrary. What we define the length of the backlink as 2 sigma of the Gaussian, and then we have this amplitude. So for several, around 100 events of backlink events, we have performed different statistics. So for example, the backlink length is around in the range of 2 to 5 micrometers, with a peak mostly in 2 micrometers. The maximum amplitude is in between 1 and 2 micrometers, with a peak around 1. So of course the backlinks, they are more long than the amplitude they have. And also because we also computed what was the initial backlink length, backlink amplitude, and of course it's much smaller than the maximum amplitude. And this is a picture in time, for example, of one event. So we observed for a long time, like almost 30 seconds, that the microtubule is almost straight, just a small amplitude. And then very soon the backlink develops, and we have a growth of the amplitude. And then at some point here, most probable thing is that the molecular motor detach, and so the microtubule relax, and sometimes it relaxes to a new amplitude that could be bigger than the initial one. And the events, we have two types of events. Some of them, when they start with a very small amplitude, they grow like exponentially in this way. But sometimes, when the initial amplitude is bigger enough, we observe, like in these simulations, that we have observed at the beginning a linear, or we fit this because it's linear at the beginning and then it's stabilized in a constant amplitude with some characteristic times. So we also, well, this is the time, the characteristic time of the buckling, and they have an exponential distribution, so mostly the medium, the average value is around 10 seconds. And this is more or less in agreement when we perform simulations in the range of 5 to 15 micrometers. Of course, we have a high variability, and we perform the simulation with 5,000 times the viscosity of water. So let me make some conclusions. So what we can conclude is that the buckling lengths are shorter, of course, than the microtubule length. So if you have a very long and deep compressed, you will observe a bigger buckling length, but we observe a shorter one. So this suggests to us that the microtubule is like anchored with some obstacles that we cannot see in the movies, and so that the effective forces, they have constraint in middle of the microtubule, not only in the tips. Well, we perform a very simple mechanical model that can simulate the shapes, the amplitudes, and the time courses of the processes, and we can also simulate and estimate with at least in the order of magnitudes their characteristic times. Of course, in this model, for example, if we have clamped boundary conditions or just free boundary conditions, sometimes the forces can be 4 times bigger. And so it's important of the role of the boundary conditions. It's also important how is the initial amplitude of the buckling and this effective length. But from the simulations, I mean, we perform simulations always in the range of 5 to 50 piconewtons. That is more or less what was estimated before from forces of molecular motors. I would also point us that the main source of a force in the microtubules is that they, in the experimental group, was doing some treatments of the cells. So this beam blasting drug stabilized this process of polymerization. So I said before at the beginning that these tubules, microtubules, grow and shrinks, but with this drug we stop this process. So with this we can at least induce if this contributes or not to the buckling processes. And it was observed that with this drug the frequency of the bucklings was not reduced. They observed that the bucklings were independent. It seems that they are independent of this polymerization process. And on the other hand, when they treat the cells with sodium azide, so this takes out the ATP, so it's like the cells don't have any more fuel, so these molecular motors cannot run. And in this case, yeah, they observed that no new bucklings were developed. So it's like the molecules, like the cell stays like freeze. So that's why we, after the simulations and these treatments of the cells, we induce that the molecular motors are the main source of active forces. That was already hypothesized, but we confirmed with these simulations. And so all this work was already published in April in European Biophysics Journal. Just shortly, some perspective. We are continuing working with Luciana Bruno, now studying the case of this constant velocity that we observe these instabilities. So we are doing some theoretical work for these differential equations can be solved for small deflections. For the bigger deflections, we should do more numerical simulations in collaboration with Florencia Carusella, that is working also in my university. And we are also working with Veronica D'Angelo. She's in the engineering faculty of the Universidad of Buenos Aires, doing some experiments with mylar filaments in glycerol. So this would be like a macroscopic model of a cell, of a microtubule in the cell. So we have the filament here, and we are pushing it on this side, and we observe these bucklings. So of course, they are dynamic. The shapes look similar, and with this model and numerical simulation, we want to study better these instabilities. So thank you again to the organizers, and thank you for your attention.